Okay, shall I start? Well and good. Okay, see, last class we discussed completely about DNA replication. So we saw how the two strands of DNA get separated from each other, and each and every strand is going to give a new strand. So we saw what were the enzymes that were required for that, and what is the process also. So what is the first process in DNA replication? First step in DNA replication. What is that? Activation of nucleosides. Then the second step, opening up of the helix. Who helps in opening up of helix? Helicase enzyme. Then what happens? Primer formation. So that's where the whole confusion of DNA replication got sorted out for us. So we got to know how the helicase is opening up the helix and how the enzyme is actually moving through the two strands, and the primer is actually coming and sitting in the 3' that is the OH end, and then the phosphate is forming a phosphodiester bond. All these things we saw. At the end of the DNA replication, we came to a conclusion. We saw that the whole process of DNA replication is semi-conservative. So who proved that the DNA replication is a semi-conservative model, and what is the idea of the semi-conservation? We'll see that in this class. It is a very, very important 3-mark question that comes in your board exam. You should be very, very sure of the basics of this question. Okay, we'll see that one by one. So semi-conservative DNA replication. The first one, you should know the meaning of the word semi-conservative. We completed with this one in the last class. Semi means half. Conservative means it's old. If fully a new DNA is going to form from a old DNA, then you are going to call it to be modern. Then DNA synthesis, DNA replication is modern. If it is fully going to be old DNA is there, from the old DNA you are going to get old DNA. Then you call that to be old DNA replication or conservatory DNA replication. But in this DNA replication, what you know is there is a old parental strand, and a new parental, from the old parental strand, new strand has been synthesized for both the strands separately. For 3' to 5', one separate strand is synthesis, and from 5' to 3', one separate strand is synthesis. So for one it is a continuous synthesis, for other it is a discontinuous synthesis. The discontinuous synthesis, the small fragments are called as Okazaki fragments. They remember that. So the old DNA strand is also kept, and the new DNA strand is being synthesized. So it is a semi-conservative model of DNA replication. Is it clear to you, children? So we are going to prove this. Who proved this? There were two scientists who proved this. One is Messelson and the other one is Stahl. These two scientists proved that DNA replication is a semi-conservative model. So we'll see what they did, what is the organism they took, what was the experiment they did, and how they proved this. We'll see. First one is they took an organism called E. coli, Escherichia coli. What organism is it? What microorganism is it? It's a bacteria. Okay, it's a simple bacteria, a prokaryotic bacteria. They have to grow these kinds of microorganisms in a medium. So they took a Petri plate, they put some medium to it. It can be a sugar medium, it can be a protein medium, the bacterial medium, in which the nutrient medium in which the bacteria can actually grow. So they took the medium, and then they added some E. coli to it, and in that medium they added a nitrogen containing medium to it. So they added the ammonium chloride to it, NH4Cl. So the medium in which the E. coli is growing actually has got NH4Cl. There is a salient feature of this NH4Cl which they took. The NH4Cl which they took had N15. It had N15. N15 means this is another form of nitrogen which is called as heavy nitrogen. It's not a radioactive nitrogen. It's not a radioactive nitrogen. I'm not going into the concepts of radioactivity. It's not radioactive. It's just another form of nitrogen. It is a heavy nitrogen. You will understand, apart from the normal N40, this is going to be a little heavier. So it is a heavy nitrogen. And the general nature of Escherichia coli is, if you put E. coli in a medium, what happens is if it starts dividing at optimum conditions. What is the meaning of optimum condition? The temperature should be there, the humidity should be there, the pH should be constantly maintained, the food nutrient availability and the metabolism of the organism that is undergoing it, all should be constant. Everything is constant, then you call that to be optimum condition. In optimum condition, E. coli will divide completely in 20 minutes. Divide completely means confluence. You remember the concept of confluence? Cellular imbibition, contact imbibition, confluence. In the Petri plate, single layer, as many cells as it forms, the one cell signals the subsidiary cells to stop its growth. That is called as confluence. So this reaches the confluence exactly in 20 minutes. So they took this, they put NH4Cl, they put the E. coli inside, and then it started to grow. And after it's finished growing, they started doing DNA analysis too. So we'll see that what they did one by one. The basic thing, the basic instrument which they used to study this was a centrifuge. A simple thing was a centrifuge. So what is a centrifuge? Can anyone tell me? Separating the components based on the density. To keep it simple, based on the density or based on their mass. So if I tell, let me suppose, see, I'm tying a stone to a thread and I'm just rotating it. Which is centrifugal, which is centripetal? Which is centripetal, which is centrifugal? Which force acts inside, which force acts outside? Centrifugal acts inside and centripetal acts outside, is it? It's completely wrong. Centrifugal acts outside. That is why it's called centrifuge. It's called as a centrifuge because the force is being applied outside and all the components are being pushed in that direction. That is why it's called as a centrifugal force. Is it clear? So they used the simple centrifuge to separate the different kinds of DNA that they obtained from this kind of experiment. So we'll see what they did. So first experiment was they took the E. coli in a medium containing NH4Cl and this NH4NS15. So what happened after 20 minutes? When all the E. coli got divided, if you take any E. coli, it will be having these two strands of DNA, and these two strands of DNA both will have N15 and N15 because obviously it has got only N15 and NH4Cl, so it will be having only N15 and N15. Is it clear? Now what they did, they took this E. coli, that is the E. coli that is formed in this Petri plate, they took that and then they put a sample of that into another Petri plate that also contains NH4Cl, but not with heavy nitrogen, but with N14. Now what happens? This DNA undergoes replication. So what happens in replication? This strand goes separately and that strand goes separately. Okay, so this is the parental strand and this is the parental strand. So whichever is going in a different way, that is 3' to 5' or 5' to 3', one has to go continuous synthesis and the other has to go discontinuous synthesis and forming octazothic fragments. We will not go into that, but by the end of the replication, it will be having two strands. This DNA will have one strand and this DNA will have one strand. Am I right? Both of them will have a complementary strand with each other. So apart from the parental strand, a new DNA strand which is actually synthesized, which is actually produced, will be N14. Because only that is available here. Am I right? This is semi-conservative. One of the parental strand is coming and the new strand is also produced. Is it clear? Now they took it to the next generation. Now they took some samples of this. It is just like the Mendelian inheritance only. They just took some samples of that and then they put it in another culture medium and then they started to grow. Now what happens? These two are parental DNA. Am I right? So the first one is these two get separated. N15 and N14 get separated. So I'll put P for parent. This is the parental strand. This is a parental strand for this. Now for this DNA, N14 and N15. This is the parental strand and this is the parental strand. And again I subject it to 20 minutes for NH4Cl and this N also has only N14. Is it clear? When this is happening, again there will be N14, 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 N14 added to all the DNA strands complementary to each other. So finally, they'll be getting completely a semi-conservatory model where one of the DNA strand is completely retained. One of the DNA strand is completely retained in the whole process of replication. Is it clear? So to differentiate between the N14 in NH4Cl and N15 in NH4Cl, I initially told you they used a completely simple method of centrifugation. They did centrifuge to understand these experiments so simply, and this is called as a CSCL centrifuge, cesium chloride centrifuge, which is sometimes what happens to increase the mobility of the substances which are there in the component, to increase the mobility. They add some chemical in the centrifuge and then they rotate it so that the mobility of these substances increases when the moisture content is more. You're able to understand, when you hydrolyze it, the mobility becomes more. So they add some cesium chloride to it and then they did the centrifuge. When they did the centrifuge, they found this N15 containing DNA of the E. coli. They sedimented at the end of the centrifuge too, because they are obviously very heavy. So they sedimented at the end of the centrifuge. And when they did the same with the second generation, now these are hybrids. Am I right? These are complete hybrids. So these hybrids sedimented somewhere above the heavy. So they were found somewhere in the middle. Is it clear? They were not found at one corner at the end, but they were found in the middle because they were hybrids. One strand was N15 and the other strand was N14. When they went to the final, the last generation which they got, the result was completely mind-blowing, because if you find there are two strands completely with N14, and here it is N14, N15, N14, N15, and again N14, N14. You're able to find that. So N15 is very heavy. N14 and N15 together is actually a hybrid. If both are N14, then it is going to be very, very light. You're able to understand, it is going to be very, very light. So what happens? They got a heavy band and then they got a light band. Heavy band in the sense they did not get a heavy band as like this. Like hybrid, exactly. Like hybrid, they got a band here. Okay. So using the centrifuge technique, they proved that there is always one strand of DNA which is always got from the parental strand. And thus Messelson and Hehel proved that DNA replication is semi-conservative. Is it clear? Fine. So I'll erase this. We'll go to the next one. There's one more. Stop one more.